guys. Thanks for coming to this event. This is the DUJS 2012 spring release event. I'm Daniel, and this is Andy. I'm the editor-in-chief, and...
Oh, yeah, I'm the current DUJS president. And yeah, I guess we're going to get started with our spring 2012 release event. So first I'd like to introduce David Bucci. Professor Bucci earned his PhD at the University of North Carolina in Chapel Hill in 1998 and has been at Dartmouth almost since then. His lab studies behavioral and neurobiological factors that regulate learning, attention, and memory. And he teaches Psych 6 intro neuroscience every winter in the psych department. And he's going to be talking about exercise and the brain. So I'd like to turn it over to Professor Bucci.
Okay. Is this good? Guy with the earphones? All right. So I'm just going to, so thank you, first of all, for inviting me. Congratulations on the release of the new issue. I just want to apologize. I have like the worst head cold in the world right now. My voice is just shot, and I got it back about an hour ago, thank God. But if I start having a coughing fit, I'm not dying. You don't have to come up and give me CPR. A bottle of water, maybe, if it's out there. But I apologize, because I probably will start coughing to death. So I'm really happy to come and talk to you for a couple minutes, and I promise it will be just that, 10 or 15 minutes. If you have questions along the way, please let me know. And I'm going to talk to you about some research that we've done in our lab with respect to the effects of exercise, physical exercise on the brain and behavior. And I just have to say, as I was driving back here, and it was downpouring, I saw, one of these like just hardcore runners, coming up schlepping up the sidewalk. And I tried not to, but everybody's splashing them with their car, and guys just drenched. And exercise is good, but you got to look at the radar too. And maybe make a choice to just hold on a little bit. But the other thing I want to say before we begin is that while this is research that's gone on in my lab, it's been actually accomplished by a whole slew of undergrads in particular. And I highlight them as we go along, and I'll point out their names at the end. Don't worry, Angela, I'm not going to embarrass you. Okay, so your brain on exercise. So what I thought I'd do is just tell you a little bit about what's going on in the field of exercise in the brain. And as you may have seen on the Dartmouth webpage recently, we've gotten a little bit of press for one of our recent articles, and I'll tell you about that tonight. Might have been written up in the D as well last week. So here's the scoop. So physical exercise and cognitive function, I think, not surprisingly, like everything else, physical exercise turns out to be good for your brain, just like it's good for your heart and your lungs and many other parts of your body. And so we knew this, and it's not brain science, sorry for the pun, to basically put these two together, right? However, I think how these two go together is what's of interest to me, and I think is of interest to a lot of people right now. So to start off, one way you could get a relationship between physical exercise and cognition is simply through angiogenesis, right? So new blood flow, new blood cells, new oxygen, more glucose pumping to your brain like every other part of your body. Sounds like a good thing. Certainly at work here. That's not what I'm going to talk about. Okay, what I want to tell you about instead are what's come to light probably really only the last 15 years or so about some very brain-specific mechanisms. Okay? And these two are not mutually exclusive and not unrelated, and I'll try to tie them together for you in a couple minutes. But this is what I want to talk about tonight. So first of all, I try to make this a combination of a little bit of serious science, but not too crazy because it's after 5. So exercise, which is abbreviated EX throughout my slides, increases this thing called neurogenesis. People know what neurogenesis is? A fair amount? Okay. So if I was standing up here about 20 years ago, people would start throwing things at me if I said, hey, you know what? You can grow new cells in your brain as an adult. And that was blasphemy. Turns out in 1965, this guy Altman realized that you can grow new brain cells, particularly in the hippocampus, but nobody believed him for quite a while until some techniques came on board in the 90s and into the 2000s. And so there's this phenomenon known as neurogenesis. It only happens in 2 places. One is the hippocampus, which is down here circled in red, and the other place we're not going to worry about tonight. But the hippocampus is a place that's very much involved in learning and memory and cognitive function. Okay? And so exercise will increase the birth of new cells in you and me. I'm not talking about infants here. Okay? So normal adults, excuse me, we have the birth of new neurons, and exercise increases that. So that's one mechanism that's been studied. Another one is that exercise increases the release of basically Miracle-Gro, thank you, for your brain. That comes in the form of brain-derived neurotrophic factor. That should be BDNF, not BNDF. So exercise increases the release of these growth factors. And again, if you're wanting a little more science, I included a picture here for you of how BDNF binds to a certain type of receptor and causes all kinds of things to happen inside the neuron, so that the neuron grows, functions better, has more synapses, that sort of thing. Okay? And so these are known. So we didn't really discover any of that, but we've explored this a little bit further. So again, not a big surprise, but the interesting story is the underlying how this happens. Exercise has great effects on cognition. This is from a review paper showing that people who exercise versus controls have better so-called executive function. So these are prefrontal cortex, inhibition, decision-making type processes, controlled behavior. So again, sort of focused concentration, controlling inhibitions, et cetera. Spatial learning, they also respond faster. Again, not terribly surprising. And I'm not saying this research isn't important, but it's not terribly shocking. I think what's really important is how this happens. So here's some current questions that we and others have been starting to address. So the first one is, how do you get from the body to the brain? And everybody's dying over here. So indeed, exercise will produce changes in blood pressure and respiration. It's good for your cardiovascular system. But how does that translate into the brain? Is it just bigger blood vessels in the brain? Yeah, maybe, but there's more going on. And so here's some areas of research currently underway. Again, this is not our work, but others, looking at endorphins, so your body's natural painkillers. And so this seems like an easy target. So you've probably heard of the so-called runner's high. So people who particularly run marathons talk about this sort of euphoria type feeling. That's your body's pain system kicking in, endorphins. And so one, I think, pretty easy conclusion to make was that, well, exercise is increasing endorphins. Endorphins are a brain nervous system kind of thing. Well, maybe that's how you get these brain effects. And that turns out not to be exactly accurate. There's a little evidence for it, but not a ton. There's some other researchers looking at interactions going on in the hypothalamus between changes in blood flow in the brain, adrenaline or epinephrine, and some of these growth factors. If you had me in Psych 6, you might remember this guy, Gribble, involved in learning and memory. One area that we are looking at are these so-called epigenetic changes. And so these are these changes, not changes to the genome that involve mutations, like we all think of, but these are a whole other class of changes to the genome that can take place in the way of chemical modifications, histone acetylations, DNA methylations, that change how genes are expressed. And this can happen any point during the lifetime. So you can change your gene expression, but it doesn't have to be through mutations, okay? So it can kind of be during your lifetime. And exercise seems to do that, right? So that's one current question. Again, nobody has it quite figured out, but clearly there's got to be some relationship between these cardiovascular, angiogenic types of effects and these more brain-specific ones. And there's probably lots of ways that they interact. So that's one question. Here's another one that we are working on a lot in my lab. So the question is, nothing against the hippocampus, and I love people who study the hippocampus, but everybody studies the hippocampus. And so our question was, could you get effects of exercise on other types of learning and memory, perhaps mediated by other structures? And so the other structure we were interested in was this area called the perirhinal cortex. Okay? And as you may know, I think as we all realize, there's different types of learning and memory. We can learn about the cues in this room or our spatial environment, or we can learn about specific things like this object, this clicker. And so the perirhinal cortex seems to be important for that latter kind of memory, object memory, okay? Not so much the hippocampus. And so we simply were curious if exercise had a similar beneficial effect on memory that took place outside of hippocampus and hippocampal structures. And so here's a rat. And rats either ran or they didn't in a running wheel for about 4 weeks. And it turns out if you put a bunch of them in a cage with their wheel, they share it, they're very nice, they play well together, they spend about equal amounts of time on there. And we gave them this very simple object memory test. So here's 2 identical objects. In this case, they were like blue Lego towers. So you get to play with Legos in my lab. It's kind of fun. And I steal them from my son, who gets really upset, but they come back with poop on them. That's all right. And so the rat explores them. That's what rats do. They like to explore things, sniff them, check them out. And 24 hours later, you give him a 3rd copy, not one of those 2, because you don't want it to smell, have him recognize it because of smell. You give him a 3rd copy, and then you give him something else, like this red cylinder. And the rat tells you that it remembers the object if it spends more time with the red one. So in other words, he remembers having seen the blue one before, so he's going to hang out with the red one. Okay? And again, just to give you a little bit of data, not push you too hard here after hours at the end of the term, but on this y-axis is basically just a ratio, a discrimination ratio. If it's above 0.5, it means the animal has remembered that object that it saw previously, and so it's spending more time with the novel one. And rats that don't exercise, the way we do the test is pretty tough. The rats that don't exercise aren't so great at this. So they're kind of around 50-50 in terms of how much time they spend with each object. But rats that exercise consistently show this little increase in object memory. So they're better at it. Okay? And down here is that discrimination ratio. And on the y-axis is amounts of that trophic factor I told you about, BDNF. And what we found is there's a very nice relationship, as just a little subset of the data, showing that if you have higher levels of BDNF in exercising rats in the perirontal cortex, you do the task better. Okay, so the answer was yes, that exercise does affect other types of learning and memory outside of the hippocampus, and the mechanisms seem to be similar. Now, neurogenesis doesn't happen outside the hippocampus, so we're not going to pursue that here. So it turns out, and here's the study that we published recently that got a little bit of press. It also happens in not only humans, but humans, guys. So we did a study with Dartmouth undergraduates who are normally fairly sedentary. And you guys actually are pretty, it was hard to find enough people. You guys are pretty active. And for the lack of a better place to start, not that we think undergraduates are rats and vice versa, but we ran the rats for 4 weeks, so we ran the humans for 4 weeks. Sounded good place to start. And so you had to spend 30 minutes on the treadmill for a couple days a week at a sort of a brisk walk. So you didn't have to be jogging, you didn't have to be running a marathon, it was a brisk walk. Okay? And we did an object recognition memory test, not with Legos this time, but sorry, this is small. The umbrella is very appropriate for today. But a watch, so that people saw objects, and they were asked to basically say, did the object appear on that list you saw earlier, or is it new? Very same sort of procedure as in the rats. And so we really wanted to see, does the rat research translate to humans? Because that's really what we care about. I'm not sure I want my rats remembering better as they're running around my basement or something. So we wanted to see if this worked in humans. And then we also looked at BDNF, the alleles for BDNF. Okay, so again, as you likely know, genes come with 2 copies, 2 alleles. And normally at the 66 position, there's 2 valines, valine. And about 70% of Caucasians, anyway, have valine. About 30% have a substitution with methionine. Okay? And so we were curious whether there's any interaction between how BDNF is functioning, and it functions better, it's more active in people who are valine than people who have the substitution. So BDNF doesn't work quite as well. We wanted to see if that interacted with exercise. And so just very briefly, I'll tell you the outcome. So people either remain sedentary, they exercise for 4 weeks, either or not, including the day of the testing. I could talk about that later if you want. Or they just exercise for 1 acute bout. So these are some controls. But here's the punchline. Oops, sorry, my little animations aren't working. So what I want to focus your attention on is that basically the people who exercised for 4 weeks and the morning that they came in and did this recognition memory test showed improvements compared to when they did the test before exercising. And what was really incredible, I thought, was that the people who were homozygous for valine, so the valine people, those were the people that reaped the benefits of the exercise. If you had met 1 or 2 copies, you did not reap the benefits of exercise. This has all kinds of interesting implications. I want to know what I am, because then I can justify not going to the gym, but that's probably not the point of the study. So it looks like exercise improves object memory in humans, Dartmouth undergrads, and it particularly helps people with the standard BDNF allele. Okay, so 2 other quick questions I'll tell you about. Does exercise affect the brain and cognition differently depending on when, like how old you are when you exercise? And so we know a fair amount in the literature about older subjects reaping the benefits of exercise. Their hippocampus volume comes back, so there tends to be a decrease in volume with age. But in adolescence, and in particular all the way up till about age 21, 22, there's parts of the brain that are really still developing and are very plastic. Okay? And so there's a lot of potential for plasticity in adolescence, and it's peaking. And notice that this BDNF thing is involved again. Okay? And so we thought, well, maybe we could capitalize on the normal plasticity by adding in this exercise brain plasticity factor, this growth factor, and get even bigger effects. And so again, just to briefly walk you through this, the bottom line is this. These are rats again who exercised as adults, and then they were tested as adults. And right after exercise, we got our usual effect. Exercise helped them. They didn't exercise, you didn't get this memory effect. But 2 weeks after you stopped exercising and you were tested, the effect was gone. However, if you exercised as an adolescent and then we let them grow up and become adults, so they were still tested at the same age as adults, I think, which is a critical piece. Sure enough, right away there was this sort of immediate effect. And it was there 2 weeks after exercise. It was there 4 weeks after exercise. In fact, we couldn't get it not there. Okay, so we seem to have these longer-lasting permanent changes if you exercise as an adolescent. And again, that seems to be related to these BDNF levels, which is shown on the right side. All right, this is the last one, I think. Another area we're exploring is, can exercise be used as a treatment for different types of cognitive dysfunction? And so I think we've all heard that exercise, if you're depressed, does wonders. Well, having had a little bit of depression in my life, the last thing I wanted to do is get out of bed and go for a run. I just wanted to stay in bed, okay? And so it's kind of hard to see how exercise could really be used for some sorts of problems. But some colleagues of ours up at the University of Vermont are interested in kids with ADHD. And as Angela knows, I'll just point you out for 1 second, who's worked with our rat version of this. There's rats who also have characteristics of ADHD. They're not ADHD rats, but they're like them. And so what I just want to show you here are 2 pieces of interesting data. Down on the bottom left, this is a task where a rat is just placed in this chamber and a light comes on. And when the light comes on, the animal rears up on its legs. He looks kind of cute. He smells the light, checks it out. Like a human or any other animal, if that light keeps coming on time after time and nothing else happens, you should stop rearing up on your hind legs and smelling the light. It doesn't mean anything. Why waste the energy? This is called habituation. And so over blocks of trials, if you see this top line here, these rats that we didn't do anything to, these are the hyperactive rats, don't habituate. They just can't get enough of that silly light that keeps coming on. They go up and sniff it like they've never seen it before. And what has been shown by a couple of a graduate student and an undergrad in my lab is that exercise will reduce this sort of excessive orienting to the light behavior, this distractibility. And again, without going into the details, all these lines grouped together here, these are lines representing animals that were treated with Ritalin or atomoxetine, which is another drug commonly prescribed for ADHD. And what we found was that exercise was just as good as some of the drugs at decreasing this type of distractibility behavior. And in kids, just a little snippet of data, so we can get them to do these sort of inhibition tasks and see if they keep responding when they're supposed to or not by having them play red light, green light, which is kind of fun. And what was shown was that there's a significant effect of exercise on inhibition. They're better stoppers, which is interesting. They also have benefits in the social behavioral realm, so they interrupt less. There's less frequent interruptions of teachers and things like that. So maybe exercise could be used. A couple other tidbits to leave you with. We and others are starting to find that there's sex differences in the effects of exercise on cognition. It's rather curious but interesting in the literature that if you exercise socially, so as a group, whether it's humans or rats, the benefits are bigger than if you exercise by yourself. The effects on cognition are much more robust if it's voluntary exercise rather than forced. So for a rat, that would be the running wheel versus a treadmill. And so this is great for me. I mean, I hate the treadmill. I hate going to the gym. I basically fall asleep on the treadmill. And so I have a great excuse. I'd much rather go out and play group sports with some friends. And maybe that makes sense for whatever reason. And we're currently looking into that. And again, I just want to point out the people who actually did the work. We got some great generous funding. We have collaborators at different places. But here in yellow, I think is the troupe of undergrads who contributed to this exercise research in the labs. These are all Dartmouth undergrads of one flavor or another, some here, some gone. Not dead gone, but gone, like graduated gone. But all of them have participated in this research. Many of our authors on the papers that this data has come from. So thank you, and if there's time, but I don't want to take too much time, I'm happy to answer a question or two.
Did you look at male and female students? Divide those up into...
We did, but we, I can't remember, I think we either had, I can't remember which one it was, we had too few either males or females to do any statistics of any real power. So we didn't really see anything that I want to write home about. But another subsequent study, we would do that purposefully. Like I said, it was hard to get students for this because it turns out people are pretty active and we needed them to satisfy these sedentary levels. I thought everybody was like me and sat around all day. But it's not. Anything else? Okay. Thank you.
Hey, thank you for Professor Bucci. Next, I'd like to, first, there's Jewel of India coming on the way, so yeah, that should be here soon. But anyways, next I'd like to introduce Professor Gribble from the chemistry department. He's an organic / heterocyclic chemist, and current projects include triterpenoid compounds as like anti-cancer compounds and antimalarials and other indole chemistry. And he teaches Chem 57 and 58 in the Chem department and some other advanced classes. And he's going to be talking about wine and winemaking and some of the compounds in wine and their biological activity. So I'll turn it over to Professor Gribble.
Thanks. This one I cut down. Okay, I got it. Is that too dark? That should be okay, I think. I've got some nice color slides, I think, so this might be better. Well, thanks, Andy, very much to you and your committee for having me. I've been making wine a long time, and actually my youngest brother in San Francisco worked in a wine supply store. He was 18. He started making wine. He's not a chemist, and his wines were fantastic. Chardonnay and Cabernet. He would win contests at the California State Fair, for example. And if he could do it, I've got to be able to do it as a chemist. And so I started in 1978 when he, in fact, had a wine supply store. So I bought some equipment and came back to Dartmouth after a sabbatical at Caltech and started making wine. So I want to talk a little bit about wine itself, the chemistry of wine, how wine is made, red and white, a little bit different, and then some of the chemistry of wine and the chemicals, as Andy said, that are in wine. Now, around the world, you need a certain number of days of sunshine. And these swaths, these areas, in fact, around the world, are where the grapes grow best. And you can see, most of the states of the U.S. actually are suitable. And in fact, every single state has at least one winery. That was not the case maybe 25 years ago, but every state in the U.S. has at least one winery. Then, of course, we have Spain, North Africa, Europe, France, Italy, Spain, Germany, of course, and South Africa and South America. Very prime areas now for, in fact, raising grapes. In Germany, the great vineyards around the Rhine and the Mosel rivers. And the rivers are very conducive to, in fact, these are all not working anymore, growing grapes, because, in fact, the cold air can slide down into the rivers, and that will, in fact, really prevent the grapes from getting too hot and also getting too wet. And the Riesling grape in Germany, very famous. This is really the most important white grape, at least in Germany, the Riesling. And you can always identify a German wine bottle by the shape. It's usually either brown or green. I'm not going to point at all. And then Italy, of course, beautiful spots, even though they're heavily involved, of course, with earthquakes, this part of Tuscany in particular. Bordeaux in France, we have the famous red wines in Bordeaux. And you can always identify, in fact, the Bordeaux wine because it has a shoulder. The Bordeaux wine bottle has a shoulder on it. And that's there, in fact, to catch the sediments that this particular grape, Cabernet Sauvignon, actually produces sediments that your parents, if they drink this kind of wine, will notice, especially on aging. We'll talk about the chemistry of that a little bit later, in fact. Decem, the famous sweet wine in Sauternes. Very expensive wine because, in fact, here the grapes are allowed to rot. A noble fungus actually desiccates the grapes, removes some of the moisture. So, in fact, you concentrate the grapes. And then what they do is they go through the vineyard and actually pick individual grapes. As you can see, the ones that, in fact, are moldy here. And then Burgundy. Burgundy is different from Bordeaux. Burgundy in France, this region uses a single grape. The Pinot Noir for red grapes and Pinot Chardonnay for whites. And again, you can see the bottles because these grapes don't throw the sediment on aging. These bottles, you can see, don't have a shoulder at all. They're very sloped. And of course, Pinot Noir was the subject of this movie a few years ago, Sideways. And it's ironic that, in fact, you can make a white wine out of a red grape because the color from a red wine, in most cases, is from the skins. The juice is actually colorless. So both a Chardonnay and, in fact, a Pinot Noir can be made, or at least a Champagne can be made, as you can see, from a red grape. Sorry. As you can see, this grape makes both a white wine, a Champagne, and a red wine. In California, very famous, where I get most of my grapes, in fact. Much of California produces grapes. In fact, Sonoma and Napa are the two prime areas, but all of these other areas now produce very nice wines. You can see all the wineries that are in Napa Valley alone, and this is an old slide. Some of the vineyards in Napa and Sonoma. And they vary in size from the smaller one. Sterling is kind of a mid-size up near Calistoga, if you know part of California. Then Gallo, which looks like an oil refinery, as you can see. And here's a famous red grape also in Bordeaux, but California as well, Cabernet Sauvignon. Now, California wines were not really considered to be good wines up until about 1976. And this was a famous wine tasting. 9 French wine experts actually tasted French wines blind versus California wines, red and white, Chardonnay and Cabernet. And you can see the top 2 wines in each category were in fact from California. And the French winemakers, when they realized this, were extremely upset. And so, in fact, they duplicated this tasting a few years later, and the same thing happened again. That a California Chardonnay beat out a French Burgundy, a white Burgundy, as you can see. And also Stag's Leap, a Cabernet, was number 1 over these very famous red wines from Bordeaux. And so after this, California wines were really respected more than they were before. And Finger Lakes, where I also get my grapes, just sort of, as you can see, west of Syracuse, as you can see here. These are the Finger Lakes, and I get most of my grapes from New York, right in this area. Lake Seneca, south of Geneva, New York, a very nice area for getting grapes, not too far away. Now, New Hampshire had a number of vineyards for a long time. I was able to get grapes quite a few years, but all of these vineyards are now gone. They're out of the picture, even though New Hampshire has a number of wineries, commercial wineries. In fact, these vineyards just couldn't make a living, basically because of the grapes they grew. You can see you get a tremendous foliage here. But in fact, the grapes are not the famous grapes of California or Europe, but rather French-American hybrids. A little bit different. They have a slightly different taste, they tend to be more acidic than the California and the grapes. Well, about 1982, actually, I started my own vineyard in Norwich. I live very close to Dan and Witt's at the foot of Bragg Hill Road in Norwich. I planted 30 vines, got wonderful growth, wonderful foliage, as you can see here. I had 30 vines, basically. I had 5 rows here, and I would get tremendous foliage, but maybe over 8 years, I made maybe 2 gallons of wine. Very difficult to get a sufficient crop. And the reason, in part, was the fact that you have a number of problems. Well, this is my vineyard, or my yard today, my vineyard so-called today. You can see it's basically gone. And the problem is you get winter kill, very cold here. You don't get the 100 days of sunshine. You get birds that love to eat the fruit just when it's ripe, of course. Insects, deer, raccoons, frost, fungal disease, and the grapes never get ripe. So it's really a no-win situation. So then I started going to New York and having grapes shipped in from California. But it was a lot of fun anyway. And this guy, of course, is a major pest now in most of the country. Okay, the vine develops actually after the wintertime. The snow will melt, bud break occurs, and then you get a rapid growth of new canes, and then you get the development of flower buds about this time of the year. In fact, heavy rain like we're having now is very devastating. And I hope in New York that the vineyard I go to in October will not be devastated because of the heavy rain they're getting actually today. Then you get flowering, and I'll show each one of these. Flowering, and then a very rapid growth where you'll eventually get grapes, in fact, through September, and then harvest late September, October. And you can see the new canes are pruned in the wintertime, and bud break. Then each one of these will be a cane, actually, that will develop, as shown here. And then rapid flowering, the baby grapes. And then in August or so, you begin to measure the sugar levels. And what you also measure, in addition to sugar, using a refractometer or taste, is the pH of the wine. You want a pH not to be too low or too high. pH typically in grapes is about 3.3 to 3.5. If it's higher than that, it's not acidic enough. The grapes and the wine then will be susceptible to really spoilage by bacteria. Then harvest time is really a very happy time, as you might imagine. And this is kind of a summary of how you go from the vine or the grapes to the finished wine. It's a little bit different depending on if it's red or white. But the vine itself, as you can see, really goes or is dependent on the variety, the climate, and the soil. And this takes about 100 days typically. Then you crush the grapes. And if you're making a red wine, what you do, you take the must, and the must is simply a collection of the juice and the skins, the seeds, as you can see down at the bottom. And then you inoculate this with the yeast, and then fermentation begins. And with a red wine, you want to have high temperature in order to extract the color and the character from the grapes. So typically the temperature can actually get up to about 85 or 90 degrees Fahrenheit in this very rapid process over 5 days here. And then you press the wine. I'll show you this in just a minute. You press the must out, and that's simply a process for separating the skins and the seeds, stems, and skins. Then you have the rough young wine, as you can see. Now with whites, it's a little bit different. Here, in fact, you take the grapes, and typically you don't ferment white grapes in the presence of the skins. You separate the skins by pressing, and then you simply ferment the juice. I'll stick to this one. And then you go right, in fact, to the young wine after inoculation. And then you have a long process here. Now, normally you will ferment a white wine at lower temperatures to capture the aromas. And so that temperature might be 55 degrees, and that'll take a little bit longer. So this is a New Hampshire vineyard. In fact, Halcyon out near East Andover on the way to Laconia on Route 11. And the quantity of grapes here is probably enough to make about 3 gallons of wine. It's typical 12 to 14 pounds will make a gallon of wine. A little bit less wine will be made from white for the same amount of grapes. And then again, in 1982 is really my first and my best crop, as you can see. This is the total crop of Deschanach, a red wine, a red grape, from 1982. Now, typically, you don't actually pick the grapes when you harvest the grape. You simply grab a cluster if you do this by hand. Many big wineries use mechanical harvesting, and you simply have a pair of scissors and simply cut off the stem, and then you crush the entire cluster of grapes. The problem with this is you have to watch out, because when the grapes are ripe, they're attractive to birds, of course, and also to insects and bees, yellow jackets in particular. So you have to be careful that on the other side of this you don't have a yellow jacket that you in fact grab, and that happens actually not infrequently. Now here's a mechanical harvester. The advantages are it's very fast, of course, and probably cheaper, but it actually shakes the vines, and so everything goes into the hopper, not just the grapes, but leaves and insects, maybe some birds as well, as you can see in this operation here. Now in Germany and parts of France, you actually pick the grapes and then crush the grapes in the vineyard. You can see this little guy is actually crushing the grapes that his father, I presume, actually dumped these into the crusher. I have a very tiny crusher, as you can see. My daughter is now 41, so you can see this goes back a ways, and a graduate student crushing these grapes here. Now, commercial wineries actually have a crusher destemmer. It turns out the stems are loaded with very harsh tannins, and you have to remove the stems when you start the fermentation. And these crusher destemmers do that. They actually crush the grapes and then actually remove the stems. The stems are ejected out at the end of this. And so the must, as it were, without the stems, simply drops down into a fermenter. Now, in the olden days, and in fact this is actually true today, you can see in Mexico they didn't have a fancy crusher, they used people. It turns out the human foot is very gentle and will crush the grape without breaking the seeds or the pips. And so you can see these guys are marching, crushing the grapes as this guy plays his harp, as you can see. And now I had a number of graduate students in those days, and they were eager to learn the operation, the process, eager to help, in fact. And before I had my own crusher, I was able to entice some of these guys to, sorry. That's a true picture, not from Norwich, not from my vineyard, but Romania or Bulgaria, I think, actually. Okay, so making white wine, again, you crush the grapes directly into carboys, as you can see. And then you test. You'll actually settle the juice, and this will eliminate a lot of maybe the skins that get through and some other things. As you can see, you'll settle this, and then you test the sugar levels, just using specific gravity, make adjustments as necessary, and then you'll rack. And racking is simply a way to siphon the juice, stay over here, the juice away from the sediment. Now, some winemakers will actually ferment all of this together. I usually prefer to remove the sediment. It makes a cleaner wine at the end. And then you simply rack it. You, by gravity, simply siphon this into a clean carboy without disturbing the sediment. Then you do testing. I have my own assistant chemist that helps me out with this, as you can see. And you do titrations. Tartaric acid, you measure both the sugar and the tartaric acid, because tartaric acid is the acid in grapes. It's the only fruit that has tartaric acid, really, are grapes. And when a grape ripens, the tartaric acid goes down and the sugar level goes up. And when you have that happy medium, then in fact, you can harvest the grapes, ideally. Then fermentation begins, and I buy a cultivated yeast. Begin fermentation, very vigorous initially, and that's where you want to have the yeast exposed to oxygen because it needs oxygen to grow and to multiply. And you can see the fermentation is really very vigorous. Then when it calms down after maybe a week at 55 degrees or so, then in fact you'll put on a fermentation lock, and this was invented by Louis Pasteur. This is a very simple device. I think I have it on the next slide. Very simple device. It allows the CO2, which is the byproduct of fermentation, as you know, to escape through the bubbler, but it does not allow oxygen to go back in. You have a water level here, in fact. So you have a very rapid fermentation, depending on the temperature, but it usually takes about 3 weeks for whites, maybe a week for reds. Then you're ready to rack. And what you normally do is to cold stabilize. And this is something that was discovered by Louis Pasteur, and he discovered optical activity, as you may know if you took Chem 57 or 51, using these crystals. And this is potassium bitartrate. And it actually softens the wine. It removes excess acid when you cool this down. You get a beautiful crystallization. And then you can simply rack or siphon the clean wine off of all of the sediment here. And you can see the result. And all of this is done without filtration. You don't have to filter the wine if you do this racking a couple of times. And this is a very nice process. You don't expose the wine to air if you use a filter. Now, red wine's a little bit different. Here you have to remove the stems. I don't have a crusher destemmer, just a crusher, but I did have a number of graduate students who were interested in learning. No, they were interested in learning the process, so they would happily sit for hours on end, and well, I would be there too, removing the stems by hand. And this is very tedious, as you can imagine. You have a cluster of grapes, and you have to physically remove the berries, the red grapes. And then the crush comes, and then after fermentation, you have the harvest. And this, as you might imagine, after they had done it for a year or two, it really took some convincing to get them going again. Oliver Hardy before he teamed up with Stan Laurel. But now, in fact, most of my red grapes, because the grapes are, in fact, better than those in New England, New Hampshire, for example, come from California. These are vineyards up in Napa. They will pick the grapes, crush the grapes as well, crush and destem, freeze the must, and then ship the must out to Massachusetts, where I belong to a co-op in Worcester. And they arrive in early December, still frozen, or at least very cold, and then you let them warm up and thaw and start the fermentation. And this is a typical fermenter now for red grapes. This is in my living room. Again, you want the temperature to get up to maybe 85 or 90 degrees for one day to extract the color of a red grape. And to do that, you actually have to punch down the cap, because during fermentation, all of the skins will rise to the top. And in order to get the color in the wine, you have to push that down. And that's punching down the cap, as it's called. And this is what's done in commercial winemaking. As you can see, here's a cap puncher, if you will. And the other technique is simply to pump over. That is, you can pump the wine from the bottom of the fermenter over the top onto the cap, and again, to extract the red color. And then the pressing time comes. Here's my son. He's now 31. You can see this some time ago, guarding my little press. Again, graduate students helping out. And it's a fun time. This is, again, after the 5 days of fermentation, you have a rough wine. Now you have to separate the wine from the skins and some of the stems and seeds. And even the chairman of the chemistry department, Peter Jacoby, actually wants to learn how to make wine. And I'll give him a few bottles at the end, of course, anyway. And this is after pressing. Again, this is a rough wine. It's still cloudy. It's still fermenting a little bit. It's very important if you get into this after you're 21, you start making wine. It's important to press before all of the fermentation is complete. Because once all the sugar is gone and you don't have the protective CO2 blanket, that's when oxygen can ruin the wine. So in fact, during all of these pressings, this wine is still fermenting, actually. It's still giving off CO2. It has maybe 5% sugar left of the initial 22%, something like that. And so the fermentation finishes in the carboys, again, protected, as you can see, with the fermentation locks. This will take maybe 3 weeks. Then you have a very nice wine. And now, depending on your style, you will wrap this into a barrel, perhaps. And then you'll age it. And red wine in a barrel will be there at least 6 months, maybe to 2 years, depending. Now, barrels do get abused. They tend to leak if they're not kept full, and they're expensive. So I use small barrels, The bigger barrels I used, you can see the problem I had. Okay, these are some of the grapes that we've talked about. These are the famous grapes. There are actually many more. There are 5,000 varieties of just this one species. And they're Native American grapes that are in a different category. But these are the prime winemaking grapes around the world. Fruit wines also make very nice wines. I've made all the wines in red and some other ones as well. And you can blend a Cabernet with a blueberry. I've won some awards with the Cabernet Blackberry Port, for example, raspberry wine. Very nice wines. And I've seen recipes, in fact, for all of these other types. Even tomato and rhubarb, things like this. Now, blending, as I just said, is really very important. You can make a rosé either per se, or you can blend a red and a white wine and make a rosé that way. One of the first wines I made from apples in my backyard, I had a crabapple tree. I made a wine, 1978, it came out way too sweet. So I blended it with a cheap French Cabernet, called it Crabinet. My mother loved the 78 Crabinet. It was her favorite wine. Or that's what she said anyway, so who knows, I guess, for sure. Okay, looking at some of the chemistry, again, you have sucrose in wine, and this is hydrolyzed easily. Sucrose is a disaccharide, as you know. It's hydrolyzed to glucose and fructose. Fructose is about twice as sweet as glucose, but both of these ferment out by a long process. I don't have a slide showing this. That would be a little too much chemistry, I guess, tonight. But all of these other sugars are present in grapes as well, and they also do their own chemistry. They can be fermented. So you get really a myriad of compounds present in grapes and then in wine as well. The carboxylic acids, as I said, tartaric acid is the major acid in grapes. Malic acid, the acid in apples, is also present in grapes, and in particular the malolactic fermentation, the secondary fermentation you may have heard about, is the process of where malic acid is converted into lactic acid. And in fact, lactic acid has only one carboxylic acid group, so it actually softens the wine. And most French Burgundies, for example, the Chardonnay, and in California, in fact, you have one of these groups, I'm not sure which one, is lost, and in fact, that will reduce the acid by 1 unit. And that's a way to soften the wine as well. But all of these other acids are there. They all undergo their own fermentation, esterification, for example, leading to really thousands of compounds are present in wine. And in fact, speaking of that, it's been found at a level of a part per 100, you have about 5 compounds. Lower levels, parts per million, you have maybe 60 compounds that have been identified for sure. Parts per billion, about 300 compounds. Parts per trillion, and that's a very small quantity, as you can see, but it's been found. All of these have not been identified yet, 2,500 compounds. And a guy at Cornell, a Cree, has estimated that there are probably 20,000 compounds that he's been able to see or estimate that are present in certain wines. Here's a Chianti wine, this red wine from Italy. You can see the wine itself has a number of compounds. All of these peaks, of course, represent a single compound. And these are the volatiles. This is just the aroma that you smell. If your parents like a Chianti, for example, that's what they're smelling. And many of these compounds will be detectable even though they're present in very tiny amounts. I'll talk more about that in a minute. Some compounds have very low or high, depending on how you look at it, odor thresholds, in fact, as you can see. So one of these compounds might be detectable by the nose, where one of these maybe is not. Let me just show you what a part per million is. Again, some of you have seen these slides. If you took 58 with me, or chem 63, very small quantity, as you can see. But it's easy to detect a level these days at a part per million. Part per billion is 1,000 times smaller, so these analogies are more impressive, as you can see, perhaps. And this is changing all the time, of course. I actually weighed a crouton for this analogy. So, now, some of the chemistry that goes on during the aging process in the barrel during fermentation and also in the bottle. Esterification reactions you've all had if you've had 58 or 52. Very simple process. Alcohols and acids, carboxylic acids, react to form esters, as you can see. You can form acetals, hemiacetals, aldehydes, and alcohols react, and this is reversible. Lactones, cyclic ketones, all these things get together, and you can have these with all the carboxylic acids I showed you. So it really leads to a fantastically large number of compounds. But, winemaking does go astray. It's not like mixing sodium chloride and silver nitrate to make silver chloride. That happens every single time. Winemaking is really different every year. That's what makes it so interesting. And you can see it does go astray sometimes. And you do get surprises. It's one of my favorite Far Side cartoons, Gary Larson. These two surgeons are operating on this guy, and whoa, this thing flies out. Watch where that thing lands. We'll probably need it, you can see. So whatever that is, I guess. And certainly luck, because again, it's different every year, and you have to be lucky sometimes in order to make a nice wine. Now, looking at some of the aroma compounds, when a wine connoisseur says that this wine smells like cherries or raspberries, he doesn't realize it, but he's actually smelling the same compound that's in cherries or raspberries that's in that particular wine. So, in fact, looking down here, Pinot Noir and cherries have in common, in fact, benzaldehyde, which is a common compound we've used in the lab, perhaps. And if you smell benzaldehyde, it does kind of smell like cherries. It's not the only compound, of course, in cherry aroma or Pinot Noir aroma, but it is certainly a part of it. Gewürztraminer is this famous compound. It's also in grapefruit and muscat wines. And this compound is amazing. You can smell this, as you'll see on the next slide, at levels of parts per trillion. And in fact, for a long time, that compound could not be detected by HPLC or GC, but your nose could smell it at levels of parts per trillion. You would need a very large quantity of wine to inject in order to see it by HPLC, for example. This methoxyisobutylpyrazine. Also, it's the compound in bell peppers. And so a wine like Sauvignon Blanc or some Cabernets from California have that compound, and it smells a little bit like bell peppers, the same compound, in fact. And here's the pyrazine. You can see, you can smell this at very low levels, and these are formed biochemically from this dialdehyde. You know, if you took 58 with me, we talk a lot about dicarbonyl compounds, very reactive because the two carbons are both electron deficient and they're next to each other. And they react with the amide of the amino acid leucine. Now, here's the amide. They just go together. As you know, amines condense with these aldehyde groups, and that's how the pyrazine ring now forms in these vegetables and in these wines. And again, you can detect this at parts per trillion. Just amazingly sensitive is the nose. Again, a part per trillion is a million times smaller than a part per million. So it's a very small quantity. Very few chemicals are toxic at this level, and very few compounds you can smell at this level. The pyrazines are one. And this is an amazing analogy if you think about it. And that's a long time. Well, your parents might like a dry martini. Now, the gypsy moth is amazing because the male gypsy moth can smell only a few thousand molecules, only 10,000 molecules of the female sex pheromone, and that might sound like a large number of molecules. But if you think about it, and you guys can do this calculation, a single drop of water has 10 to the 20th or 10 to the 21st molecules of H2O. So molecules are really, really small. And so this guy is just amazing. His antennae here are able to detect only a few thousand molecules of sex pheromone. Okay, some of these wines are actually, or some of these compounds in wine are present in fruits, and isoamyl acetate is banana oil. It's kind of a noxious smell. In fact, you may have made this in the laboratory. It's also present in some wines, and it's the honeybee alarm pheromone. So you never want to go on a picnic near a honeybee hive with bananas and some wines, especially rotten bananas, because, again, this is the alarm pheromone of the honeybee. And this amide is the alarm pheromone of the yellow jacket, and this is known to be present in Zinfandel, this red wine from California. Now, these are the wines, or the compounds, the anthocyanins, that are responsible for the beautiful color of fruits and certainly red wines. And depending on the groups attached, as you can see, the color varies a little bit. Now, on aging, I mentioned this before, on aging, a red wine will actually change its color a little bit, and in part, it's undergoing this chemistry. Here's one reaction that has been identified. As you can see, it's reacting with pyruvic acid to form this compound, which is now a little bit less bright red than the original compound, as you can see. And when these things get large enough, they precipitate out, and this is the sediment I talked about at the beginning that you have in red wine. Some wines can live a very long time. It really depends on the temperature of storage and also the corkage, how sound the corks are. Here's an 1870 Lafite from Bordeaux that was tasted 100 years later by this famous wine connoisseur, Broadbent. Here's another wine from 1799, a Lafite, again in Bordeaux, tasted in 1960. And it's still a little bit faded, as you can see. But Chelychev actually put this into perspective. He tasted in 1898 and 1971 that you have to have some imagination, perhaps, to really be honest about what you're tasting. 1784, you can see this wine from Dechem, very expensive wine. Again, was tasted in 1985. Just amazing, 200 years old. Still pretty good. Now, when a red wine ages, as I alluded to, it loses its color, actually gets more brickish, more brownish. A white wine darkens, and eventually they actually will approach and become almost the same color. And here's a Dechem, which is a white wine. Originally very old, as you can see, 1825 or so. And this is maybe just 10 years ago. You can see it's almost a red wine now, or an old red wine, if you will. Now, what about the health effects? You've read a lot about these. These polyphenols really have tremendous antioxidant properties in preventing cancer, heart disease. And one of the mechanisms for these polyphenols, if you will, that are present in fruits and vegetables and wine is that they react and decompose the diol epoxides that we talked about in Chem 58 and Chem 63, if you took that with me. These diol epoxides of benzpyrene are actually destroyed very rapidly. And of course, these are potent carcinogens known to react with DNA. Resveratrol has been in the news a lot lately. We're still not sure about the aging potential of this compound, but it's ubiquitous in fruits and vegetables and also wine. But there is evidence it tends to lower the bad cholesterol and, in fact, increase the good cholesterol. And you can read about that. Yeah, I'll be glad to send these slides to anyone if you'd like. I know they're recording this, but if you'd like a copy of the actual slides, just let me know. And heart disease as well. You can see it tends to block the oxidation of the bad cholesterol into the plaque that will clog the arteries. Wine is a little bit better than other forms of alcohol, but alcohol itself actually has these benefits within moderation, of course. Here's a study out of Denmark. I'm just about done. Out of Denmark, where you can see the more wine you take, the longer you'll live. Mortality risk goes down. With beer, it tends to fluctuate, but it is pretty constant. Hard liquor, you can see, tends to increase after the number of drinks. This is quite a bit. So there is a difference between, let's say, vodka, whiskey, and wine. And again, a study showing a consumption of heart disease, wine consumption in heart disease. You can see France is much better than the U.S. They drink about 10 times the amount of wine that we do. Now, fruits have these antioxidants as well, as you've read about. Certainly blueberries and cranberries, and all these fruits have antioxidants. So you might say, well, why wine? Why not vegetables? Why not peas or cranberries? Why do we have to drink wine? Well, there is a difference, and the difference has to do with the fact that the polyphenols in wine are dissolved. They're soluble. They're not in the vegetable matrix. And so, in fact, they're more available, these polyphenols that are in wine already, as you can see. So why alcohol? Why not just alcohol-free wine, which would have the same number, presumably, of the antioxidants? Well, here again, the alcohol, because the alcohol can dissolve the polyphenols, in fact, they're more available in wine than in alcohol-free wine, which, in fact, is available. So some final quotations, going way back, as you can see. Plato, Pliny the Elder, Louis Pasteur, of course, and Ben Franklin as well. Yeah, no. Okay, finally, I do have a quiz for you guys, because you're all scientists, I know, or budding scientists. I'll give you some clues. Okay, I know you're hungry, so I'll go right ahead. I was born in San Francisco, never saw snow, so had to do something with all the snow in my driveway. So I'll be glad to answer any questions. Nice. Well, the estimate had to be from either GC or HPLC, where you could actually count all the little blips. And then I think what he did, he probably took a portion of that, expanded it, and then counted more, and then just assumed, multiplied, and estimated 20,000. Yeah. But there probably now maybe 3,000 compounds have been identified in wine per se, lots of different types of wine together.
No more questions then? Okay. Well, thank you for coming. We have before we break and you guys can eat Jewel in the back. I'd just like to draw your attention to our spring issue. So the theme for this issue is extreme science. It's not a very particularly specific theme, but there's a lot. It's given us some flexibility to explore some really cool topics. And so I encourage you guys to pick up a copy. They're in the back over there or outside, and take a look through it. So thank you again, Professor Gribble, for coming.
Thanks
and
very much.
Thank you to Andy for arranging this. And yeah, so food in the back. Great.